Many thanks, Garan, for this nice introduction, and I also want to thank him and the organizers giving me the opportunity to present here some of our data in this form of this webinar. And the title you already can see here on the first screen. So it's CRISPR-Cas-mediated mutagenesis of plant traits, and this is performed mainly at my previous employer at IPK Gattestheim. I moved to HHU in August 2020, and since then I'm establishing the Center for Plant Genome Engineering. And since November, I'm also associated scientist in the CEPLAS, the Center of Excellence in Plant Science, where several universities and research institutes belong to. So to give it a start, so this, as I mentioned, so at Heinrich University, just recently the biology institutes moved into this new building, and our center is located here in this building, and we have facilities which allow us in order to produce and analyze genome-edited plants. And to give it a start, I want to start with the challenges which we are facing. So I'm sure you all are aware of, on one hand, we have an exponentially growing world population. So, and this, of course, is accompanied by a demand for producing more food. But at the same time, what we also observe is a severe change in climate. And this is accompanied by the fact that, for example, also fungal diseases are spreading and increasing. So this means that plant fungal communities appear in areas where they usually do not exist. And as genotypes or varieties are usually locally adapted, this does not mean that they are prepared for these fungal infections. And so therefore, farmers and breeders are facing problems in order to find a way to adapt their breeding material to the new challenges. And what are those kind of breeding targets? On one hand, of course, it's yield performance. So, of course, the farmers are interested in getting out as much as possible from their fields. Next would be, of course, becoming more and more important, yield security. So as I mentioned, so on one hand, we have problems with biotic and abiotic stress factors, which of course influence the yield dramatically, and also the changing weather conditions. Then, of course, one of the breeding targets is flowering time. So I briefly mentioned that, of course, we are using locally adapted material, and this means that for the same species, you have to adapt the plants to the local requirements. And one of those is, of course, the flowering time, which influence. Then, of course, one of the breeding targets is fruit or seed color. So since we are mainly working with cereals, so of course it's the seed color, which is some examples given here. And during domestication, also the farmers and breeders selected for this trait. And of course, one of the targets is also fruit and seed composition, so like gluten content of wheat and so on and so forth. Yeah, and others apply as well. So how the breeders can cope and in order to achieve. So here on this slide, you can see the development of the breeder's toolbox. starting with the Angian breeders, they started just with the selection breeding. So, and later on, they started crossing, which of course allow just to feed a smaller population of people. So then later in the 1900s, hybrid breeding started, which is called also the first green revolution, which was also due to the demand of a growing world population. Since the 1980s, we have a so-called biotech breeding, which includes also the generation of genetic engineered plants. And last but not least, so we have now more biotechnological approaches and tools in the box, like marker-assisted breeding and also genome editing or gene editing, I have to say. And over the last 2 decades, there are several platforms developed, which are listed here on this slide. So on top, you see zinc finger nucleases and transcription activator like endonucleases, talents, and zinc finger nucleases are protein-based endonuclease platforms. So usually they consist of 2 parts. You have one part which is responsible for the binding to the DNA, and then you need a scissor. And for these 2 platforms, it's mainly the FOC1, and as this is working only as a dimer, so therefore you need also 2 halves of your platform. And these are 2 protein-based, so also the binding to the DNA is based on protein or peptides, and you have your scissor, while the most recent development in this field are the clustered regularly interspaced short palindromic repeats, short CRISPR and Cas9 associated protein. Here you have also 2 components, but in difference to the 2 upper, this is based on an RNA and protein complex. In this case, the RNA guides the scissor to the target site. And if you want to modify a gene or a certain given genome, of course, there are some requirements. And one, of course, is to know the sequence. And here, the advances in sequencing technologies, which lead to the next generation sequencing platforms, allow now even the analysis and the knowledge of rather complex genomes like those of barley and wheat. But as these are huge data sets, of course, you need also developments in analyzing and processing this. And so therefore you need also a strong bioinformatics, which allow the analysis of those huge data sets in order to get sense out of it. And then last but not least, of course, you need also tissue culture that you are able to make out of a transformed cell a new plant. So there are several forms how those gene editing dysfunctional units can be transferred into a cell, for example, a plant cell. And so there could be plasmids or linearized fragments. It could be in the form of RNA, and it can also be as a protein, like for the CRISPR-Cas in a ribonucleoprotein complex. And then, of course, you need also to transfer those functional units. And for this, usually we have 2 platforms. On one hand, it's viabilistic gene transfer, and on the other hand, it's Rhizobium radiobacter, usually known as Agrobacterium tumefaciens. So the biolistics, which also become more and more again important, has the advantage that it's host more or less host independent. You do not need binary vectors, which make also cloning a bit more complicated. It can deliver DNA, RNA, and/or protein. But on the other side, it leads to complex transgene integration pattern, which is, of course, not, you don't want to have if you want to make stable modifications, because then on one hand, it could lead to gene silencing, or if you want to inherit your integrated DNA, then of course, this leads to complications in order to get homozygous material. On the other hand, the rhizobium radiovector, this is a natural process which we make use of. And so this is mainly host dependent. We can only deliver DNA, but it's mainly used for stable approaches, and it has the benefit of simple transgene integration, which allow an easier inheritance and stability for the expression. But recently, there are also other platforms and technologies which are in use for gene editing. So like electroporation, PEG-mediated techniques, or more recent, the use of nanoparticles. This could be silicon-based or arginine nanoparticles, nanoclothes, cell-penetrating peptides. So there are a number of emerging technologies which allow the transfer of our functional gene editing units. So coming now to CRISPR again. So this is a natural process also, which is adapted from a bacterial immune system, so from Streptococcus pyogenes. And usually in the bacterium, it's used as an immune system, as I say, in order to avoid infections with bacteriophages or plasmids. In biotechnology, we use just two components. We use the RNA and the Cas9 protein, which is the scissor. So what all the platforms has in common, that they induce a double-strand break. And as these double-strand breaks are also occurring naturally by environmental factors, the cell has developed over the evolution several mechanisms and pathways in order to cope with those double-strand breaks. And the two main pathways are listed here on this slide. So they are non-homologous end joining and homology-directed repair. The majority of cases is following this non-homologous end joining pathway because it just sticks together the two loose ends, and so it's therefore not a lot of effort. And because of this just stick together, so then you can expect that there are some error, and so this technology is error prone, or this pathway is error prone, leading to insertions or deletions. So what would be the outcome of such an induced double-strand break? So here you see not the DNA, you see the messenger RNA and then the protein. So if you alter the DNA, of course, you will alter your messenger RNA and subsequently your protein. So as I told, following the non-homologous end joining pathway, you can have either insertions or deletions of single or several nucleotides. And this usually, as we read these triplets for one amino acid, leads either to an altered amino acid sequence or to early stop, so which will abolish your protein. So we started early on in adapting one vector, which is illustrated here. So Stefan Lieke adapted this vector, and it consists of the OSU3, so the rise U3 promoter, you can use type 2S restriction enzymes to include one gRNA, and we have a codon-optimized mice codon-optimized Cas9 protein, which is also under the control of the mesovikinin promoter. And in cereals, we prefer to use the hygromycin selection because this avoids escapes. So here on this diagram, I summarize some attempts for 11 target sites. And what you see here on the x-axis are the 11 targets. And on the y-axis, we have the number of plants. In the light green, you see the number of regenerants. And then the dark green show you the number of mutant plants which was observed amongst those regenerants. And as you can see, so we have quite good, but also in some cases less efficient mutant frequency. To give you some examples of published work, so this is a collaboration with the group of Niels Stein and Minju Lee, postdoc in his group, and he confirmed the candidate gene for the famous barley albostrians mutant. So it has been described already in the 1950s, but until a few years ago, the responsible gene was not known. And it was famous because it gives this variegated striped leaf phenotype. And the group of Niels Stein identified this gene via tilling. And so the tilling mutant contains a 4 base pair deletion here in this position. And we used CRISPR-Cas9 in order to give another confirmation. And as the Cas9 protein needs a so-called PAM sequence, which is MGG for the Cas9, we could not directly target this tilling position, but we choose target sites which are close to the tilling mutant. And so what you see here below is the outcome of those attempts. So we got regenerants which has a mutation in this gene, and so these pictures here show already T1 plants of the primary transgenic, and so you can see that we could confirm that this is the causative gene for this phenotype. So a second approach which has been published together with the group of the late Petr Galushka and Veronique Bergenou. So here Katarina Holubova was studying some hormonal composition in barley, and we were targeting cytokinin biosynthesis in targeting the CKX1 gene, which is a cytokinin dehydrogenase. And they started 2 attempts. On one hand, they used RNAi technology, which are here symbolized as knockdown. And together with us, we generated knockout plants. And these are highlighted here in the rather red color and called knockout. And what you can see here is over the development of a plant, cytokinin activities. In gray, you see the wild type, and in blue and in red, you see the transgenic mutants, either knockdown or knockout. And as you can see, so there is a statistically significant reduction in the cytokinin content in those plants. And we can also observe, also not statistically significant, but we can still find an increased cytokinin content in the grain. Another outcome of this site-directed mutagenesis could be that you delete codons. So this means that you have a deletion of 3, 6, 9, 12 nucleotides. And so therefore, you can alter your protein sequence without abolishing. So you just take out one or several amino acids. And this can be also used for the discovery of functional domains. And again, in collaboration with the group of Niels Stein and Ming-Ju Lee. Again, so we studied the homologous gene of albostrians, which is the CMF3 gene or albostrians-like gene. And so here we had also, the group of Niels Stein had also one tilling mutant, which has here a stop codon, and we also selected guides which are in close vicinity to this. So if you then look for the protein sequence of some of our mutant plants, so we have some which are still wild-type-like, but we have also individuals which has an altered protein sequence. And luckily, we could see a difference in the phenotype. So the presence of this domain leads to different chloroplast development. And so thereby we could identify, so there was already 4 functional domains described for the albostrians, but by doing this site-directed mutagenesis, we could confirm that there is another functional domain amongst those genes, which is highlighted here. Cindy Schedel, a former PhD student and postdoc, also in the group of Jochen Kumlin at IPK, when she was establishing the site-directed mutagenesis in tobacco, she observed some similar phenotypes. So she made use of the green fluorescent protein in order to establish this mutagenesis. And so here you see the GFP wild-type sequence and the target site. And of course, so you would expect a bright fluorescence. And so then amongst the mutants generated, she observed some individuals which had modified protein sequence here at this position 130, which completely abolish GFP fluorescence. And as this is within the center which is responsible for the fluorescence, so this was known before. Another observation which she made, and which is also related to what I have told you before, so by deletion of 3 nucleotides, she loses just 1 amino acid, which lead to a so-called attenuated protein function. So you could alter the protein of investigation, you can weaken, or depending on the outcome, maybe even find something different. And the same holds true for another mutant. So this has also a -3 deletion, but since it's at a different position, so here we have not only the deletion of 1 amino acid, we have in addition a change of 1 neighboring amino acid, which also lead to a different fluorescent outcome. So the take-home message here is that you can use this technology also in order to make proteins which has an attenuated protein function. This would be somehow similar to the RNAi approach, but would have the benefit that it's genetically fixed and you don't have to overcome a certain threshold to start the silencing effect. So here you can also, or for lethal genes, by doing this, you can also may find some viable plants. Another application of this technology has been also illustrated in the group of Jochen Kumlin by Nagu. So they made use of the fact that you can use white hybridization so that you can fertilize egg cells of one species with pollen of another species. And the benefit of this is that during or after fertilization and cell division, you get rid of one of the genomes. And so this could be useful if you have or if you want to do site-directed mutagenesis in a species which is rather recalcitrant for transformation, like wheat. And so here Nago used wheat, maize, pollination in order to induce mutations. So how does it work? She generated transgenic maize plants which harbor the Cas9 construct which is targeting a wheat gene. So within the maize, it will not induce a mutation. But if she has this now fertilized to an egg cell of wheat, so then during the zygote and the further development, you will induce a mutation in the target genome, while afterwards you get rid of your transgene. So this has several advantages. On one hand, you will lose the transgene already in your regenerants. On the other hand, one transgenic maize plant can be used for dozens of different genotypes of recalcitrant species like wheat, and so she successfully showed that this is true. So she could induce mutations in bread and in durum wheat, and she could also show that you can use this for several accessions or genotypes in order to induce mutations. And this is a cool technology which can be used to circumvent the problem of transformation. So coming to two more applications. So the first is dealing with so-called de novo domestication. So as I mentioned in the beginning, so the farmers started early on in selecting plants which has favorable traits like seed size or fruit size, color, of course also the seed shattering or fruit abscission was one of those domestication traits people were selecting for. And the domestication of barley was took place in the so-called Fertile Crescent, which is given here in green, so covering those countries, Jordan, Syria, Armenia, Iran, Iraq. So in this area, the domestication of barley took place. And scientists have discovered over the years several genes which are related to some of those domestication traits, which are given here, and they are distributed amongst the 7 barley chromosomes. And I will give you one example for the BRS1 gene. So the BRS1 gene is responsible for the switch between 2-rod and 6-rod barley. You might be aware that in barley we have these 2 spike phenotypes. You could have either 2-rod or 6-rod barley. And Takao Komatsuda from Japan identified the BRS1 gene as the causative gene. And in collaboration, we first started also with the RNAi technology to confirm that the gene he has selected is responsible for this phenotype, because he discovered that a functional BRS1 gene will avoid the fertility of the lateral spikes. So it's a negative regulator of spike fertility. And mutation or downregulation of the BRS1 lead to the 6-rod phenotype. And so as soon as the CRISPR-Cas technology became available, of course, I became interested in order to repeat our RNAi experiment. And so here you see the genomic structure of the BRS1 gene. It consists of 3 exons. and the target site was chosen in exon 1. So below you see then the outcome of the sequencing, and so you can see that we found mutants in this plant, and of course we checked for the phenotype, which is given here. So we used Golden Promise on the left side, which is a 2-rod barley genotype, and by inducing mutation in this gene, we could get a 6-rod barley. So we removed the awns for better visibility, because also you would have awns on all of these grains, so like it's illustrated here. But for better visualization, we use this. So by making one tiny cut and mismatch repair here, you can, in theory, triple the amount of grains per spike. The second application is more related to another use of barley as for beer. So here also we have the corona restrictions, so maybe beer gardens and restaurants, at least here in Germany, are closed. So, but you may still remember that a freshly tapped beer over time don't taste as in the beginning. And so, of course, you are interested in knowing what is the reason for that. And if you look on the biochemical background, so you can identify that this is due to oxidation, and it's the oxidation of linoleic acid by, so the final product is given here, the trans-2-nonenal, and this makes this off flavor, this cardboard flavor of beer due to the oxidation of the linoleic acid. And so the first enzymes in this oxidation process are called lipoxygenases, and in barley we have two. We have lipoxygenase 1 and lipoxygenase 2. And the two feed into two different pathways. So lipoxygenase 1 goes into this direction, and lipoxygenase 2 is going into jasmonic acid pathway. There are several functions for LOX proteins described for plant species. They are involved in several processes, but the main process we are interested here in this study is for the beer quality. On one hand, as I already described, for the flavor, and on the other hand, it's foam stability. So, and as we are a UK-based webinar, so you may prefer a beer without foam, while I prefer a beer with foam. And by just interfering with this one gene, we can use the same, more or less the same background, and can make beer for UK or for German markets. And so the LOX1 gene consists of 7 exons. So here we also choose our target site in the exon 1, and we got mutants in our regenerants. And if you quantify this, so then you can see that we have a prominent D3 mutation. And if you wonder why is this, so this is due to this repeat of GCT here at the moment, which most likely use another pathway which I did not mention before, which is the microhomology mediated repair pathway, which might be responsible for this. So we went on, and as all our primary mutants are usually chimeras, so we use embryogenic pollen cultures in order to fix our mutation and to come to double haploid plants. And then, of course, we were making powder of our grains, and then did some LOX activity measurements, which is illustrated here in this diagram. So on the left side, you see that for the wild type, we have a quite high activity, while for 5 of our DH lines, we have less than 10% activity. And for the brewing process, this is still fine because this is still called as lossless barley and would be fine. So some of you may know that Golden Promise, our model cultivar, is mainly used for whiskey production, but I told you something about beer. So of course, this would be not a useful genotype. So we repeated our experiments with some elite barley material. And I don't want to bore you with more sequencing results. We went on, we made DH, and we made multiplication because we wanted to go further on in processing and see the outcome. And so we teamed up with Martina Gastel from the Technical University of Munich, and she used our barley grains in order to come to this stage here to make some beer. And also most of the beer produced was used for some analytical analysis. So we still had the chance to get at least one bottle. And so also it looks rather from the glasses and also from the color like sparkling wine, but the taste was beer-like. And so I promise that soon you can read some publication about this project. Last but not least, so I'm almost done. I just want to come now to the Champions League of this targeted mutagenesis, which is the homology directed repair, because so far I just talked about the deleting more or less randomly nucleotides, which is of course not editing in a deeper sense. And so therefore it would need a repair template in order to really write any given genome. And so this is hardly achievable because of several reasons. And again, Nagu some years ago started to establish this at IPK by using also a simple approach, because this GFP and YFP proteins differ just in two nucleotides, which leads to one amino acid, altered amino acid, which leads to the shift in the fluorescence. And so we used transgenic barley plants, which has a single copy of GFP, and used at that time also TALENs and the CRISPR-Cas technology in order to make those kind of allele conversions by providing the GFP-specific scissors and a non-functional YFP repair template. And so we could confirm that in barley it's possible, which is illustrated here by these yellow cells. So we could switch a GFP into a YFP, and we confirmed this also by this Lambda stack that it's really shift in fluorescence. And now we did also additional molecular analysis to confirm that we really could achieve this allele conversion in barley. In a later project, my postdoc Martin Becker here illustrated or used then RNP complexes in order to achieve the same outcome. He also used this transgenic barley, but this time GFP-specific RNP complexes with the non-functional repair template. And as you can see here also, he succeeded also in this in epidermal cells. And if we then quantify this, so we usually use a co-transformed cherry plasmid as a control in order to normalize our data, because we used, as I told, GFP fluorescence. And of course, by the bombardment per se, we can also get non-functional cells. And to have a measure for still viable cells, we co-bombard the M-cherry, and this was then set as 100%. And from this 100%, so 82 was GFP negative. This means they had a mutation in the GFP gene. So 35 had a positive signal for YFP, And if we then split up this, so barley is a diploid organism, so it could be theoretically, you could have one GFP, one YFP, or one mutated GFP, one YFP, or two YFP, and this is given here. So he found that 27 of these 35 had this diploid non-homozygous outcome, but in 8% of those, he could confirm that he has in both alleles this allele conversion. And so he went on also to do this in stable transgenic plants, but this is not part of this talk here. So with this, I come to the summary. So I hope I could convince you that we established site-directed mutagenesis. I did not show data for all this year, but we have established in barley, wheat, corn, tobacco, and tomato. So the technology can be used for gene function analysis and discovery of functional domains, or you can use it for attenuated protein function. So white hybridizations can be used for targeted knockout of hard to transform species. I showed you that the knockout of VRS1 convert 2-rod barley into 6-rod barley. We had an efficient site-directed mutagenesis for the LOX1 gene, and we used embryogenic pollen culture for mutant dissolution and fixation. We could find a reduced LOX activity, and we could transfer the knowledge from the model cultivar golden promise to a lead material, which finally even would used for the brewing process and had some altered malting characteristics. And with this, want to acknowledge, so on most of the slides I already mentioned the people involved in this. I thank the Weber Lab for helping me in establishing the center here at the HHU. Of course, the Kumlin Lab, where I have been for nearly 20 years, and most of the work I presented here has been done there. So, of course, big thank to the Kumlin Lab. I thank for the funding. I, of course, also thank the Planted, which is also co-host for this event here. And we have some collaborators, of course. And I thank you for your attention. And of course, I would be happy to take any questions.
That's great. Thanks so much. That's great. Very interesting stuff. Thanks, Goetz, for a nice introduction, and then coming through some published and some unpublished again. So I encourage everyone, if you have any questions, please do write them into the question box, and I will relay them. We have a few questions. So let me first start. So a few years ago, we had the fortune, I guess, of tasting some gene-edited cabbage at a meeting in Sweden, and unsurprisingly, it tasted exactly the same as normal cabbage. So what about the beer, that the LOX minus beer that you made? Was that similar to normal beer, or was there a crisper tang to it?
No. So the taste was it was a beer. Yeah. So it was not really different tasting. Of course, the color I presented here looks a bit strange, but this might be also related to the microbrewing which has been done. So of course, this is not a fully running brewery. So also they have all the stuff to do the individual steps for this microbrewing. But yeah, so it tasted like beer.
Okay, and in those elite varieties, did you cross out the Cas9 as well? So it was so non-transgenic, I guess, at that point.
Yeah, so we just choose those segregants which lost the tDNA due normal segregation, and of course continued with those ones which are transgene free. Yes.
Okay, cool. Okay, so we have some questions. So first question from Juan Fernandez, and this is about the HDR that you mentioned towards the end. So he says, what strategy did you use for HDR? Do you use ribonuclear particles and single-stranded DNA or double-stranded DNA? Could you elaborate a little more on that strategy?
Yeah, so of course, there are several reports on the benefit of single or double-stranded DNA. So in our experiments, Martin so far could not confirm the benefit of the single stranded DNA. Also, I have to admit that the number of experiments is still limited. Yeah, so he could show, as I also illustrated here, that we are able to make this allele conversion. And this was based on double-stranded DNA, but we were also playing around with single-stranded DNA, but this was not successful so far in our hands. So far, the data I showed here are based on double-stranded DNA.
Okay. I think we have a few questions coming in about that same sort of technology. So let me ask, so a question from Steve Jackson from Warwick. He asks, have you got any experience using cell-penetrating peptides, liposomes, or nanoparticles to get your CRISPR-Cas9 RNA and RNPs into the plant cells?
So we started already many years ago with the cell-penetrating peptides, since it was published in Triticale that they even could transform pollen, which was also, of course, one of the topics in the group of Jochen Kumlin. And so we also became interested in transferring at that time just GFP in order to see if we are able to transfer something into barley or wheat microspores. And so far we failed in using the cell-penetrating peptides in transferring something functional into the cell. So we used also electroporation, and this is also advertised, but it also does not work under the conditions we tested. Yeah, so of course I don't say that it's not useful, but at least in our systems it was not working. And very recently Martin also started with nanoparticles, and at least he could transfer some protein inside of the cells. So these are the good news. So this is also working in the IPK lab. And of course, we will continue on this using nanoparticles as well. So we do not have any experience with liposomes so far.
Okay. So a question from Fabienne Nogoy. So have you tried base editing or prime editing in barley?
So in the group of Jochen Kumlin, of course, the base editing was also one of the topics, and there is also some success, but since I was not directly involved in this, that's why I did not present here. So we have confirmed also base edited barley plants, and for sure this will be published in a reasonable time. Yeah, so there is success also in barley on base editing. We did not start it yet with the prime editing in barley.
Okay. So we got a bit of a basic question here. From Andrea Todd asking about the online tools that you use to design guide RNA sequences. Which ones would you recommend for that?
This is of course also, I think, depend on the people. So there was one platform which I personally used quite frequently, which was TestGene, which was also, I think, from Cambridge, some outsourcing. But at least to my knowledge, recently this platform is no longer working. So I did not check in the last weeks, but when I had some attempts in order to reach this platform, it was no longer available, which in my hands was quite good. So there are other platforms like CRISPR or CRISPR, or I can't mention all. All has their advantages and disadvantages. Of course, if your target genome is there and you can select the guides with the genome in the background, that you can also check for off targets. This, of course, would be a plus. Yeah, so this is available for most of the platforms which I'm aware of. Otherwise, there are also other online tools which you can use for this analysis. So I would not have their favorite. Yeah, so this might be personal preferences. So this was even amongst my colleagues. Yeah, so everybody preferred a different one. As we all succeeded in selecting quite efficient guides, and of course these are just predictions. Yeah, so the Destrine one was also based on the Dönch papers from the Dönch algorithms from 2014 and 2016, where they analyzed in mammalian cells for the effect of a certain nucleotide in a certain position. And so if we compare it with this, so in part we can confirm what we have found with the mammalian cell system. Of course, over the years, we also accumulate our own data where we see now which guides we selected, which features are beneficial for the species we are working with.
Okay, so that was a question aimed at the very start of the process, but a question from Jonathan Napier, which is at kind of the end of the process as we have it. So he asks about what the situation is with field evaluation of gene-edited crops in Germany at the moment. And as he said, without that possibility, without those field trials, it's going to be difficult to translate anything, as you showed about the golden promise example. Obviously, a question very close to Jonathan's heart with his work that he does at Rothamsted on gene-edited crops.
Of course, and so you are in the lucky position that you have your protected site, yeah, where you can, because it's still considered, even if it's transgene-free, it's a GMO, and also all politicians will say that we can, of course, perform field trials after application. So the scientists are more or less not really willing to do this much efforts because of the bad experience in the past that those fields were destroyed, and you have a lot of effort and not a real outcome. Of course, there would be options outside of Germany, yeah, so that we could go to the UK or to Switzerland, and also in Russia. There might be options for gene-edited plants to make some field trials, and we are in discussion in doing this, because we would be, of course, interested in doing not just greenhouse experiments, and there's even an ongoing discussion if we should continue also with those kind of field trials in Germany. So it's still under debate and theoretically possible. Yeah, so if I make the application for such a field experiment, of course, it will undergo the evaluation, and theoretically it can be done, but it's a lot of effort with a very uncertain outcome. So of course, I would love to do also field trials, and we are going in this direction. Yeah, so we will use some of our plants also in field trials, maybe not in Germany.
Okay, right, yeah, challenging indeed. So another question from Fabienne. So he says, you mentioned MMEJ events of repair, and do you have an idea about the ratio of MMEJ to NHEJ repair in barley?
In this publication, which I mentioned for the allele exchange between GFP and YFP, so in this we had an efficiency of 3% of the mutated cells had this allele exchange, which was at that time, and to the best of my knowledge, quite efficient. Yeah, so our plant species has less efficiency. So in this case, maybe we get some credit for working with crop plants instead of models. So here the barley behaved quite well. So there are other studies which we have done also with Ingo Schubert, and so this was also in this range of 1 to 3% of the mutated cells could have this allele frequency in stable plants. Yeah, so not in, so on the cell-based level. So this, as I showed you, yeah, for the work with the RNP, so it was higher. But this is a cell-based assay. Yeah, so of course we want to have inheritable mutations.
Okay. Okay, so question from Vladimir Nekrasov, and he asks, how did you manage to deliver or co-deliver your RNP and DNA repair template via biolistics?
So of course this is still challenging, yeah. So because the DNA you can easily precipitate, and with the RNP complex on top, yeah. So this needs some technical tricks to do properly. But this would be the end of the talk in this direction. So I will not talk more in this direction. It's possible, and it has been published. Yeah. So other people in maize did it as well. Yeah. So they shown that you can do it. It's a bit tricky, but you can do it. Sorry for not being more specific. More specific. I'm sorry.
Fair enough. So a question from Jordan, and they ask about the temperature used. Have you tried varying the temperature of the plants after transformation, and whether that works differently with different types of Cas9s that you've used?
So yeah, thanks for this question as well. We are of course also aware of this temperature dependency in the systems we used. We do not see a dramatic effect of temperature. Also, I have to admit that we so far did not check too many different Cas proteins. We compared Cas12a and Cas9 in this case, and it was not that dramatic effect. Of course, we are aware of this, and there are some experiments ongoing in order to see if we can increase the efficiency by this temperature means.
Okay, cool. So a question from Johannes Stutman, and he asks, do you have any data on the HDR frequencies when inducing either with SSBs or DSBs, either nicases or nucleases?
So far, we did not use nicase for this kind of approach. So these experiments are ongoing, and this is in the pipeline. But what I have presented here is the normal Cas9 and the Cas12a. They are not nicases. So both domains are functionally, we get double strand break rather than just nicking one DNA strand. So this is also part of the ongoing experiments, but I can't present something here at the moment.
Okay. So I think we'll finish with one more question. Another one from Juan Fernandez, and he asks, do you know, or do you have any evidence if single-stranded DNA fragments get physically integrated into the genome after HDR? And do you have any data on that?
No. So I do not have data on that, but I also would not expect that a single-stranded DNA become integrated. So, but we did not check for this that much so far. Yeah.
Okay, great. Okay, so there's a few other questions, but I will pass them on to you. Good, so you can answer if you wish in your own time. But I'm going to say thank you for everyone for attending, and thanks especially to Goetz for his presentation today. I mean, it's an exciting time for this technology, and as Jonathan's question alluded to, hopefully it's technology that we will get to use across Europe and in the UK over the coming years, indeed. So as I said, so everyone, this presentation will be online. I'll put it online tomorrow, if that's all right with you, Goetz, to share this, and
Yes.
I'll share that link around so anyone who wasn't able to attend today can watch the recording of that. So thanks so much for everyone for attending, and again, thanks so much, Goetz, for your presentation. That was really great.
Yeah, many thanks for you, and I appreciate that I could present some of our data, and many thanks for all the people listening to this talk this afternoon.
Great.